Yeah.
We'll do our best.
You just flick through like this, Brad. Are you happy to do that?
I'm happy to do this. Yeah. Does this work?
You can see this, they can see this.
Sure. Hopefully everybody can see this. Okay. So let's start at the top. Okay. You should be grand rounds. Here's your code 121522. Reasons for optimism in Alzheimer's disease. We'll have about half as many reasons as planned, but we'll be good. A variety of disclosures. None of the companies listed here have anything to do with the work that I'll be presenting today. Most important slide, of course, is this acknowledgement slide. Many thanks to the people in the lab who have done so much work. Great appreciation to our funding sources, of course, and to the Alzheimer's disease research leadership. And of course, those of you who were able to see John earlier, John was the founding director of the Alzheimer's Center. So if you do a PubMed for Alzheimer's disease, there's over 170,000 articles. And having read each and every one of them, I can tell you that there are a few facts that really, are non-controversial. The first is it's a dreadful disease. It affects millions and millions of Americans and people worldwide. And now there is incredible reason for hope. Who gets Alzheimer's? Well, everyone on the planet. There are really no epidemiological data to suggest that there's any spared populations or any populations at higher risk. Just about everybody, 5% of the population age 65, 1 in 3 individuals by 85. We do know a lot about who gets Alzheimer's though, and it depends a lot on who you chose to be your parents. So at this point, you should go back and you should choose your parents wisely. Because if your parents have Alzheimer's disease, you have about a 3-fold higher chance of getting Alzheimer's disease. And if your parents are spared, well, of course, you're in a better bucket. Well, what goes wrong in Alzheimer's disease that causes dementia? Well, Alzheimer in 1906 described 2 pathologic lesions under the microscope using a simple light microscope with a little mirror in the bottom reflecting the sunlight up through the slide. And he saw these neurofibrillary tangles and these senile plaques. We now know that these are made primarily of the molecule tau, and the plaques are made primarily of the molecule A beta or amyloid. And so I'm going to use these terms more or less interchangeably, but as I go through the data in the presentation, you'll see that there's some nuances of what forms of tau are important and presumably what forms of A beta or amyloid are important. Alzheimer's disease doesn't start one morning. In fact, it begins maybe 15 or 20 years before clinical symptoms occur, with the initial deposition of amyloid plaques, the accumulation of neurofibrillary tangles. These accumulate to a point where, in fact, plaques have already reached pretty much of a plateau well before clinical symptoms occur. Tangles continue to occur, clinical symptoms progress, and finally a dementia syndrome that we know as Alzheimer's disease emerges with concomitant atrophy of the brain in specific brain areas that are referable to the clinical symptoms. So the underlying questions that I'd like to address today in order to achieve effective therapeutics are, first, which lesions underlie cognitive impairments? It seems like that's an important thing to understand if we're going to fix the cognitive impairments. And then second, is something that really emerges from seeing patients over the years, is that some patients have, well, no one has a benign course of Alzheimer's disease, but certainly some individuals have a milder course that goes on for a decade or more, and where patients from year to year don't change that much. And other individuals just have an awful, awful, awful course in which they go from nearly cognitively normal to severely demented within a year or two. At pathology, those two individuals look exactly the same. And so what's really different between them? And I'll foreshadow the changes that I'll highlight here. So it seems as if differences in the properties of the proteopathic seeds, that is the form of tau, which forms oligomers and is microscopic, meaning even smaller than microscopic, that is, you can't see it under a microscope, are important and correspond to the rate of progression. So it's something other than neurofibrillary tangles. That the genetic landscape, especially including apolipoprotein E, which is the major genetic risk factor for Alzheimer's disease, is important. And then I'll close with just a few thoughts on understanding resilience, because there is the very, very rare circumstance that's been highlighted by Teresa Gomez-Isla and Matt Frosch, in which individuals have a brains at pathology that have tangles and plaques of sufficient quantity to easily meet the diagnostic criteria for Alzheimer's disease. And yet clinically, we're either normal or darn near normal. And so what makes those individuals resilient? And I think that's a future direction that we're going to learn from. So 2 lesions to start with, plaques and tangles. Plaques are distributed across the entire cortical mantle pretty much evenly. And as I said, have reached a maximal amount in the brain even before the earliest clinical symptoms occur. However, it's implicated by genetics that A beta or plaques are critical causal in the disease. And yet there's this curious mismatch in a clinical pathological correlation sense of where the plaques are and where the symptoms are, highlighted perhaps by the fact that the medial temporal lobe, which of course gives rise to most of the amnestic early symptoms of Alzheimer's disease, are relatively spared from plaques. However, A beta is an unequivocal genetic driver of Alzheimer's disease. The autosomal dominant genes, APP, PS1, and PS2, which lead to clinical syndrome that starts in the 40s, oftentimes. These are genes that were cloned initially by Rudy Tanzi here at the MGH. So critically important and absolutely definitively linked to Alzheimer's disease. Even that common apolipoprotein E gene, the E4 allele, increases risk by 3 to 10-fold, has a dramatic effect both on the age of onset, and now it's about 70 or so for a 4-4, as opposed to about 10 years older for a 3-3, and maybe a 50% increase in the number of plaques. Down syndrome, similarly, a 50 to 100% increase in the number of plaques. So more plaques, earlier, worse disease, unequivocal over multiple causes. And I urge you to attend Grand Rounds on January 12th, when Mike Irizarry will be presenting the data from the lecanemab study, which is the first absolutely definitive study showing that an anti-A beta therapeutic has an impact on the clinical progression of Alzheimer's disease. So A beta is important, but not closely linked to clinical symptoms. And so for the moment, I'm going to put it in the parking lot, and I'm going to focus for the rest of the talk on the things that are most closely linked to the clinical symptoms. And of course, this foreshadowing says that those are neurofibrillary tangles, or more accurately, the tau molecule, which makes up neurofibrillary tangles. By contrast with the plaques, the distribution of tangles matches clinical symptoms. So medial temporal lobe, temporal pole, association cortices, and the temporal, parietal, and frontal areas severely affected by tangles as the disease progresses, with a hierarchical distribution with increasing tangle deposition in the same order as patients develop symptoms as the disease progresses, with relative sparing of the primary motor and sensory strips, which of course matches the clinical picture. So we set out to understand what it was about tangles that caused neural system dysfunction. And through the rest of the talk, I'll be combining work done in mouse models and work done in human patients. The mouse models are unbelievably informative and allow you to do manipulations and obviously experiments. But it's my deep belief that unless it correlates with what happens in people, what we're really learning about is mice. So our assumptions are that tangles cause neuronal dysfunction. Well, they line up. Tangles cause neuronal death. I didn't mention this, but the areas of atrophy in the brain and neuronal loss are the same areas that develop tangles. And that tangles look the same in all cases of Alzheimer's disease, and therefore tau is the same in all cases of Alzheimer's disease. So those are the underlying assumptions that we set out to test. We developed a technology called multiphoton imaging, which is sort of a fancy microscope in which you can put a living mouse underneath the scope and look at the brain as the mouse is sitting there, either awake or lightly anesthetized. And you can image the neurons that are there, the neuronal activity using calcium indicators and so forth. And so you can ask what really goes wrong in a mouse that neural system-wise that is developing either tangles in the 4510 mice. Slide over simplification. This is actually a P301L mutation that's driving the tau aggregation. The 22-21 mice, these are mice that don't develop fibrillar tau, but have every bit as much soluble tau as these mice do. So they're matched. And these are mice that we made in collaboration with Karen Ash at University of Minnesota. And then of course, wild type mice that just have normal tau. And when Mark Bush compared the neuronal activity of fibrillar tau mice, non-fibrillar tau mice, and wild type mice, he found something really quite intriguing. This graph shows the number of firing that a neuron in the field of view does over the course of a minute in this case. And on average, it turns out that in a wild type mouse, about 10% of the neurons don't fire in any given minute. They will fire the next minute, but any given minute that you're monitoring, about 10% of the neurons are silent. And that's these control mice. In the 21-221 mice, about 40% of the neurons don't fire in any given minute. And that persists over time, of course. And the same is true in the 4510 mice. So these two mice, despite our hypothesis that this fibrillization process was the critical thing that would lead to neuronal dysfunction, in fact, like many times, it's the control experiment that's really important. And the control mice had the same phenomena. Now, both of these mice were engineered so that we could turn off the transgene when we wanted to. You do so in this instance by feeding the animals doxycycline, which is just a molecular trick. It's not a therapeutic. And when you turn off expression of this continued soluble form of the gene, both the 21-221s and the 4510s recover to wild type, so that the presence of this fibrillar tau actually doesn't impact the ability of the neuron to fire. Well, they're mice. So it's a weird thing, but they're mice. However, it resonated with me because many years ago, well, not that many years ago, when Teresa Gomez-Isla was a fellow, she looked at the superior temporal sulcus of human cases and found that neurofibrillary tangles accumulated with increasing duration of disease, as we expected, and neuronal loss accumulated with increasing duration of disease, as we expected. But there was this odd discrepancy between the number of tangles and the amount of neuronal loss. In fact, a discrepancy of over 10-fold. So that the amount of neuronal loss, although it correlated with the presence of tangles, didn't really add up right. And we've actually replicated this now in those same 4510 mice and in 2 other mouse models. And the same thing occurs, that the amount of tau aggregation and the amount of neuronal loss, there's this 10-fold discrepancy. So we have just much more recently developed a technology, and this is truly a royal we. This is Ted Zhuang and Rachel Bennett, and you heard Rachel give a wonderful grand rounds just a few weeks ago. And in this set of experiments, we were able to label neurons in the brain with one fluorophore and develop a technology to develop tangles, to label tangles with a different fluorophore. And then we could image the mice 1 week to the next week to the next week to the next week, so that instead of taking snapshots like we do with neuropathology, we could actually run the movie. We could actually see what happened over time. And so one surprising thing that fell out right away was that from 1 week to the next week, and just look at this purple box, from this week to this week, 2 more tangles appeared. So tangles appeared quickly. So although we had in our minds that in humans, it takes years to develop Alzheimer's disease, and even in mice, it takes 3 or 6 month epochs. In fact, for the individual neuron, this aggregation of tau into a tangle occurs very quickly. And in fact, in some other work that we've done, we've shown that it happens in the course of 2 hours. So bang, you have a tangle. All right, so what happens to the neurons? Well, as I said, we could also monitor neurons, and in these mice, we know that neuronal loss occurs as the mice age. Cross-sectionally, in this snapshots, there are fewer neurons in an old mouse than in a young mouse. But now we can actually watch that. So again, a neuron here, a neuron here, a neuron here, a neuron here, no neurons here. And so this neuron is lost sometime between this week and this week. And so we can actually do this for a big field of neurons. Roughly 1,000 neurons are monitored in a series of mice from week to week. And these are the preliminary results. So we were able to clearly document in the course of these experiments 64 instances where a neuron was lost from 1 week to the next and actually not present in the following week as well. So we're trying to avoid technical problems there. So 64 different rare events that occur. And of all of those neurons that disappear, maybe one of them had a tangle. And of all the neurons that had a tangle, so far, none of them have died. So that doesn't mean that a tangle won't eventually kill a neuron. That doesn't mean that if we did, 1,000 times more, that we wouldn't find the occasional. But the bottom line is that neurons with tangles, at least, are more likely to survive. And neurons without tangles are more likely to die. And that suggests that it's not the fibrillar aggregation of the tau that is causing neuronal dysfunction and death, but in fact, something about a species of tau which is being made by the transgene and is accumulating in these neurons, but is not fibrillizing. So pausing here, our assumption tangles took years to form. Well, no, a couple hours. Tangles cause neuronal dysfunction. Well, no, it seems like it's a different form of tau. Tangles cause neuronal death. Well, maybe it's a different form of tau. And now finally, the tau is the same in all cases of Alzheimer's disease. Well, I guess you've guessed by now that the observation is going to be different than the assumption. That in fact, although morphologically tangles look pretty much the same from case to case, biochemically they're not the same. So to explore this in patients, Anastasia and Romain and Simone Dujardin in the laboratory developed a series of techniques in which we could isolate the fibrillar forms of tau from these soluble oligomeric forms of tau. Most tau in the brain is monomeric, that's physiologic tau that's stuck in microtubules. This isn't going to be further considered for the moment. This form is about 1% of all soluble tau. And of course, these fibrils are what make up the paired helical filaments or the neurofibrillary tangles, the aggregates that Alzheimer could see under the microscope. So through a series of biochemical techniques, we could differentiate these, isolate them, and then ask whether or not either of these things aggregated. We could tell by mass spec with Judith Steen whether or not they were the same molecules. We could look for toxicity and so forth. And I'll just give you some highlights from those experiments. Now, this complicated slide results is the mass spec data from Judith Steen's lab over at Children's. And the top bar here is the oligomeric soluble species. And the bottom bar is the filamentous paired helical filament species. And you can see that overall, the pattern of these two is the same. Each one of these bars represents a phosphorylation or acetylation site, a post-translational modification on tau. There's gobs and gobs of them. There's about 40 of them in the paired helical filament form, maybe 20 in the soluble form. Overall, the patterns are pretty much the same. There are far more here. And then there's individual sites that are more likely to be present on the paired helical filaments than on the oligomeric soluble form. So this suggests that this form is not just a broken up part of this. It's not just a technical artifact. And it also suggests that this has plenty of changes in it that could account for different physiology compared to physiologic tau, which has only 5 or 6 post-translational modifications on it. And now here I've illustrated the same sort of approach with the oligomeric versus the filamentous tau above or below the line for just three different cases out of this series of cases that we did. And you can see just by glancing at the patterns here that the post-translational modifications from this Alzheimer case and from this Alzheimer case, although again, the patterns are similar, the number, for example, in this case, far fewer than in this case. And so we were able to ask, well, does that make any difference? Does the number of post-translational modifications, does the exact type of post-translational modifications, does the pattern of post-translational modifications impact the pathophysiology of those tau molecules? We did so by developing a technique in which we could essentially ask, how likely is it that those molecules will aggregate into an oligomeric or fibrillar form? And when we measured that quality in the tau from each of 32 different cases whom we had followed here at Mass General and whom we had in our brain bank, and Matt Frage verified had Alzheimer's disease. And we knew clinically and neuropathologically that the only disease that they had was Alzheimer's disease. So these weren't individuals who also had concurrent something else. When we looked at just in a rank order, the amount of what's referred to as seeding, the ability of those differentially modified tau molecules to aggregate versus the rate of progression that we observed clinically, we actually got a rather strong correlation, and roughly 20% of the variance in progression amongst patients could be accounted for by the exact quality of the tau protein in their brain. So this suggests that there's different strains of tau that different individuals have, and of course has therapeutic implications. Now we were heartened to see it about a year later, a group at Cleveland Clinic who looked at rapid progressors versus standard progressors of pure Alzheimer's disease, used a different set of biophysical assays that came to exactly the same conclusion, is that individuals with rapid progression compared to individuals with ordinary progression in their hands had a substantially different biophysical properties for the tau, and entirely analogous to what we saw. And now a third group in Europe has replicated the data as well, and yet a third set. And so this has rather rapidly, over the course of just a couple of years, gone from, gee, that can't possibly be true, all tau is the same, to, gee, everybody knows that, how are we going to deal with different strains? And that's still an open question, I think. So at quick summary, are the plaques and tangles that Alzheimer described in 1906 really the critical issues? Well, the lecanemab is directed against protofibrils, which don't form, which aren't the amyloid positive plaques. And our data suggests that it's the oligomeric forms of tau rather than the fibrillar form of tau, which is most critical. Patients have different rates of progression, in part because of these tau species, perhaps. But what else impacts the rate of progression? I'm going to just, as quickly as I can, cover the genetic landscape and resilience, and then really think about what that means in terms of therapeutics. So there are about 70 known gene risk factors for Alzheimer's disease. Many of them uncovered here at Mass General and Rudy's group. Most of them are very, very, very small effect sizes in the border of about 1 or 2%. But apolipoprotein E, which is present in 3 common alleles in the population, has a whopping effect, has a major effect, has a 3 to 10-fold increased risk. 10-fold for 4 fours, 3-fold for a single copy of 4. And importantly, in work that we had initially described in 1993, and then more recently, Alberto Serrano-Pozo and Sudesh Das have taken forward, E2 is profoundly protective. That I don't think we have a single E22 case in our 2,000 brains in the brain bank. And I recently did a national search to see if I could find a 2-2 Alzheimer case and came up empty. And so if you're a 2-2, if you chose your parents really, really, really wisely, you are in great shape from the point of view of Alzheimer's disease. And that sort of anecdotal evidence is shown here in this graph that Eric Ryman put together, and we collaborated in the study. And here it just is so dramatic that if you're a 4-4, you have about a 50% chance of getting Alzheimer's disease by early 70s. And if you're a 2-2, well, you never actually get to the 50% point by the time you get to be 100. And this is a clinical diagnosis. And so we don't even know whether or not these few individuals who are 2-2s actually had Alzheimer's disease or had a mimicking syndrome. So if you're an E4, you have a much earlier age of onset. As I mentioned, you have a lot more plaques. Your rate of progression of disease is actually a good deal faster. This is work that Alberto has taken the lead in. And if you're an E2, you don't get the disease to start with, but if you're a 2-3, maybe, rarely you do. And even then, if you're a 2-4, rarely you do. And even then, your rate of progression is slower, although the 2-4s are very small numbers, and so that data aren't so clear. So what's driving that difference? And one of the things that we think is that APoE4 appears to predispose to a more inflammatory profile of gene expression in microglia than comparable E3 and E2 individuals. And the E2 seems to provide a different phenotype for microglia. So there's something about the neuroinflammatory milieu of the brain in which plaques are being deposited that impacts both the number of plaques and the other neuropathologic features of Alzheimer's disease, as well as the clinical rate of progression. And Sudesh Das, who's a bioinformatics expert here at MGH, did this beautiful study in which he looked at the glial transcriptome in extant public domain data, and now we have replicated this work with some recent single nuclear RNA-seq data. So it seems like if for your brains, being an E2 would be a good thing. And as I said, choosing your parents wisely would be helpful, but it's too late for me to have chosen my parents wisely. And I suspect the same is true for all of you in this room. So can we make E2, can we reverse that decision? Can we give you E2 instead? And in a universe where gene therapy is all the rage, where you can get gene therapy for your retinal disease, and you can get experimental gene therapy for Parkinson's disease, and gene therapy is on the market for forms of ALS, well, why not? So we explored the idea that we could do gene therapy to introduce APoE2 by collaborating with Bev Davidson at Children's Hospital of Pennsylvania, and Rosie Jackson's taken the lead in all this work. And Bev has developed a vector which specifically transduces the ependyma, the lining of the ventricle of the brain. And we have taken advantage of that in order to have APoE2 expressed primarily in the ependyma. APoE2 is a secreted protein. It then diffuses throughout the cortical mantle. And we could ask, what happens if we do that to a mouse model And we chose to use a mouse model of amyloid deposition because the biggest phenotypic difference between human APoE 2, 3, and 4 is the number of plaques. So we decided to go for the biggest phenotypic difference. Work that Rosie has taken the lead in, injected APPPS1 mice, which develop plaques, which are shown here in these sort of magenta-ish colors at 4 months of age. 8 weeks later, so only 8 weeks later. And remember, it takes the virus 3 or 4 weeks to actually transduce the appendable cells and start to express it at a reasonable level. So this is probably on the order of 5 or 6 weeks of treatment. We assess the animals. And the results, I think, are really exciting. This thioflavin S staining, which is histochemistry for amyloid plaques, vehicle control animals, these animals are genetically engineered to get lots of plaques. The highest dose animals, it's an obvious difference. By biochemistry, this same thing occurs. If you measure the amount of A beta in either the soluble or fibrillar forms, again, you get a marked reduction after exposure for just a few weeks to APoE2. And this is on a background where these mice have been genetically engineered to be producing human APoE4. So this is a mimic of giving someone with established disease who already has plaques in their brains and who's a genetic 4/4, highest at risk, a little bit of E2 on top of that. What's perhaps the most exciting result, however, is illustrated in this slide. In the vehicle animals, as in all cases of Alzheimer's disease, microglia in the immediate vicinity of plaques, shown here with an IBA1 stain, are highly activated. And this is just a morphologic assessment of microglia, but it's how neuropathologists have been judging microglia for, what, a century, probably. So these are very activated microglia. And then in a dose-responsive way, just 5 weeks later, you can see that this marker of neuroinflammation has essentially, gotten better. That we have changed a very inflamed brain into a very quiescent brain from the point of view of the microglia. And this is not exactly what we anticipated. We thought that it was actually the expression of the APoE in the microglia based on the single nuclear RNA-seq data. But always, the alternative was that the APoE wasn't acting on this because the cell was making it, but was acting because a different cell made it and acted on the cell. And in fact, this means that exogenous APoE2 is able to change the phenotype of microglia. And then this is just a beautiful final confirmation that APoE2 is doing something good. In the immediate vicinity of plaques, in the so-called halo of plaques that Tara Spires in the lab described some years ago, there's a loss of synapses in both humans and in these mouse models. And that's reflected by staining with PSD95. And when you give the animals APoE2 just 5 weeks later, that has largely normalized. So the number of synapses lost in this space, that we've encouraged neuroplasticity and synaptic regrowth. So neurodegeneration, marked by the synapse loss, neuroinflammation, marked by the microglial activation, and of course, amyloid deposition, all improved over the course of just a few weeks of exposure to E2. So we're pretty excited about that, and we are actively going forward. In fact, just yesterday, I met with the FDA, who said very encouraging words. And so with any luck, we're going forward with this. Now, I'm going to just close. I know we just have a few minutes left with a few thoughts about resilience. I mentioned earlier that Teresa Gomez-Isla and Matt Frosch have been studying these cases in which there's a very obvious clinical pathological mismatch. Lots of pathology when Matt looks at the brain, very few clinical symptoms when Teresa has seen the patients or reviewed the medical records. How can that be? At a superficial level, that's again an argument that the things that Alzheimer's saw under the microscope weren't always causal of dysfunction, and there were other things going on. Teresa has done a series of experiments now showing, again, that the inflammatory environment in those brains, marked either by microglial activation histologically, by neuronal loss, or in this instance, by a whole series of cytokines and so forth, various neuroinflammatory markers, differ substantially between these resilient cases and the average individual who has Alzheimer's disease. And I think that this is incredibly exciting and brings us some hope that even individuals who already have the plaques and tangles of Alzheimer's disease, who are already in that far end of the graph that we talked about, whose brains are already affected by Alzheimer's disease, that there might be some therapies that would be. So there are 4 takeaway messages. And I'm sorry I've rushed through this a little bit, but maybe I can still summarize here. Amyloid is implicated by the genetics as a causative feature of Alzheimer's disease. And although it doesn't anatomically or quantitatively match closely with cognitive impairment, it still is an obvious therapeutic target. And lecanemab recently showed reversal of plaque deposition with a modest change in the rate of decline over 18 months of treatment. I earlier pointed out that Mike Irizarry is giving grand rounds on January 12th to really drill down on these data. Many of you will remember that Mike Irizarry is an MGH resident and MGH fellow, MGH faculty, and was in the lab for many, many years. A wonderful individual, and I'm so looking forward to having him come and visit us in a couple of weeks. This is the first data that robustly shows an effect on changing clinical progression in Alzheimer's disease. And although the effect is modest, it is the first time in 20 years we've really seen an effect. And it is in large part, I believe, because Michael and his colleagues designed a really, really, really thoughtful and carefully performed study to be able to see what was going on. I emphasized, however, at the very beginning that amyloid accumulates 15 years before the clinical symptoms. And so maybe the point to really intervene in amyloid deposition is early in the disease, in the pre-symptomatic stage of the disease, much like we intervene in cholesterol deposition and heart attacks by giving anybody who has a high cholesterol level a little bit of a statin. We don't wait for them to declare and have a heart attack first. And so is there a role in prevention? And again, MGB leads the way. Risa Sperling and the A4 trial, which has been going on for some years now, and even more recent A345 trial is testing exactly that hypothesis. And we'll know within a few years whether or not there is an effect in preventing the development of clinical symptoms. Second take-home point. Soluble proteopathic tau seeds, these oligomers that I kept mentioning, they're bioactive. We've identified their post-translational modification landscape. They correlate with the rate of progression. They're doing something that makes neurons unhappy. This implicates soluble rather than fibrillar forms of tau as the most bioactive, and therefore suggests that we have to tackle this problem at the point where tau is being synthesized, rather than after it has already had all these post-translational modifications and aggregated into a form. And in fact, I think this is just emphasized by these recent data from Ted and Rachel that fibrillar tau may not be the proximate cause of neuronal death. We refer to this form of tau as all the tau we cannot see, but it's there nonetheless. Well, Sarah DeVos, whom many of you recall, was a postdoc in the lab for many years. During her graduate school work with Tim Miller at WashU, worked with Ionis to develop a antisense technique and demonstrated that tau reduction was able to prevent neuronal loss in the same animal model that I was presenting to earlier. More recently, we've been doing some work with engineered zinc fingers, which are sort of a precursor of CRISPR-Cas. And again, we can demonstrate very, very positive effects in mice by producing the gene therapy approach rather than an antisense approach, a way to knock down the tau expression. Now, Ionis has gone forward with Biogen to test this. Their recent phase 1 data suggests that it's safe, that it lowered tau in the spinal fluid by up to 50% just 8 weeks after the last dose. And in fact, I just learned earlier this week that MGB has been won a position as a site for the phase 2 trial of the antisense medication, the anti-tau antisense medication. And so presumably first or second quarter of 2023, we'll be able to offer our patients the opportunity to have an antisense against tau. The third take-home point, I emphasize that genetic influences are critically important and highlighted the role of APoE, which is the largest genetic risk factor in Alzheimer's disease. We have been working hard to satisfy all the things that the FDA has asked us to do in order to launch a gene therapy clinical trial here at MGH. And this might provide a way to restore both synaptic integrity and dampen neuroinflammation in the brains of individuals who have Alzheimer's disease. And then I was so excited to see this just at the Clinical Trial on Alzheimer's Disease conference a few weeks ago. And this actually is the report of that conference from the New York Times. The New York Times says a promising trial targets a genetic risk for Alzheimer's disease. Front page headline. And this was a trial run out of Weill Cornell with Lexeo Therapeutics, Ron Crystal. They've exposed 2 patients by AAV to APoE2. The technology that they're using for the delivery, we think is, sort of the decade-old version of AAV2, and we believe that we've, have version 2.0, which will be much more effective at delivering APoE2 with this technology. But 2 patients, nothing bad has happened, so better, not a lot of patients, but that's wonderful so far. And already they reported some positive improvement in the CSF biomarkers of A beta tau. And so we are super excited that gene therapy might be coming to Alzheimer's disease. Then the fourth take-home point, where is the future? And I really believe that understanding resilience, understanding the role of inflammation, understanding the complex role of vascular biology, as Rachel Bennett highlighted at Grand Rounds a few weeks ago, these are all going to contribute to neural system failure. And as we learn more and more about those phenomena, we'll have more and more targets. So I just want to thank you. It's really the first time in 30 years that I have felt incredibly optimistic that we're going to have a therapy, not just for mice, because we've gotten really good at curing mice with Alzheimer's disease, but actually for patients. So thank you. Merrick, you have the floor.
I had to go as fast as I could before Andy.
No, go.
First of all, amazing body of work and presentation. I'm just so excited. I feel like we should make sure not to ask you to do anything except for cure Alzheimer's for the next couple of years. A lot of our patients, when they hear all this great work and they hear all this work in prevention, they say, well, what about my father or mother who have it now? And I was intrigued by your showing that maybe there was some plasticity. I just wanted your thoughts on, is there a repair pathway, and is there a point where it might not be too late, I guess?
So I believe, as as I've already shown you, all of my assumptions that have proven wrong when we've tested the data. So you have to take this with a little bit of a grain of salt. But I believe that plasticity phenomena are entirely possible, and that the neurons that are remaining in the brain, that dead neurons don't think, fine. But the neurons that are remaining in the brain, at least in the animal models, we can restore them to normal function with appropriate therapeutics. And I think that there are, although there's a lot of atrophy, there are a lot of remaining neurons in most of our patients. And there's probably a point too far. But I believe that the vast majority of our patients, we might be able to benefit.
Thank you.
Brent, this is just a crazy idea and maybe not square on point. If I heard you correctly, you said that fibrillar tau has an uncertain role, whereas soluble tau appears to have a much clearer role in disease progression. Would there be a?
That would be the anti-textbook version of yes.
Yeah, no, but I think I heard you correctly. So would there be a therapeutic opportunity to try to drive polymerization of tau, to try to move the tau from the soluble phase to the fibrillar phase, and basically use it as a sink to suck up this poisonous stuff?
So that's a really interesting possibility, and it's something we've kicked around a little bit. It seems likely that tangles are not good for a neuron, and you can think of it sort of as a toxic waste dump. That if there's toxin all over the community, that's really bad. If you kind of put it all together into one little area, it's still not good for the community. It might be better than widely distributed, but it's still not great. So but experimentally, it's really intriguing, and you wonder if you can sort of wall it off. And in fact, if that's what the cell is trying to do, that this is a cellular response to these misfolded proteins, is to either throw them out of the cell, put them direct them to the proteasome or autophagosome, or cause them to aggregate. And I think it may well be a cellular defense mechanism. John?
You showed us that the tau neurofibrillary tangle follows increased levels of amyloid in the brain. Is there a similar relationship to the toxic pre-tangle form? In other words, does amyloid drive your new species of bad tau?
So in experimental animals, the presence of amyloid clearly is synergistic with the presence of tau, and it makes the phenotypes worse, from formation of oligomeric tau to seeding ability of that tau to ultimately neurofibrillary tangle formation. And so there is a synergistic effect, and we don't understand what's driving that interaction yet. Kevin.
So Brad, you point out there are like 40 sites for post-translational modification of tau. And it seems a little scary to suppress the production of tau altogether. I mean, it's pretty important to cellular protein. But what about looking for genetic evidence that some of those post-translational sites are problematic or might be causing problems, and that if you track that down by mutating the tau so that it can be modified there.
So
Objective forms of, or
yeah, so it's an intriguing idea, and in fact, it's the basis of John Dixon's current K Award. So hopefully 5 years from now, when John and his team have managed to do the site-directed mutagenesis of different patterns of those post-translational modifications and mimic them in various ways, we'll be able to answer that on sort of a biophysical basis and then back it up towards clinical. But we just don't have the fundamental understanding of what any of those things do to the conformation yet. And ultimately, it's going to be the conformation of the protein that drives its interactions.
So there isn't any information that the human polymorphisms that there might be.
So human genetics just isn't informative here, which just means it's not informative in this instance. Steve.
Really cool and exciting talk, Brad. From a biomarker standpoint, is anything known about mapping the either number or distribution of post-translational modifications of tau in the brain versus the plasma tau biomarkers, current ones, future ones?
Yeah, so plasma biomarkers are an exciting field that's really just emerged in the last maybe 2 years, and plasma phospho-tau seems to be pretty robust and reliable diagnostic now, and it's moving out of the lab into commercial entities and I think soon into clinical practice. the technology for those assays is essentially always an ELISA of one sort or another. So it's a sandwich ELISA with a capture and a single epitope domain that's recognized. And so depending on which company it is, which lab it is, which whatever it is, they believe that their phosphoepitope is the most informative. And there really haven't been deep head-to-heads, and there's going to be a lot of technical issues about which assay works better just technically versus which assay is actually biologically more informative. And at least our data would suggest that it's not a single epitope, that it's going to be some pattern of phosphorylations, for example, that introduce negative charges that allow a different conformation of the protein. And so it might be that some epitopes will just in practice be more informative than others, but I don't think we're quite ready to call that yet. Yet on the other hand, it's moving so fast that maybe a year from now, I'll be able to go, look, the data show clearly it's 217's the winner, and that'll be amazing.
Are there generally plasma samples available from people during life who had the postmortem brain assays for the modified tau species?
Well, we have something on the order of 100 cases that we followed in life who have come to autopsy, where we have serial plasma samples. And as we speak, Steve Arnold and his team are doing the phospho-tau antibody tests on those blood samples. And then, of course, it's a little bit of work to get the mass spec done on the brain samples. But we're going to try to connect the dots.
Great, so maybe there'll be an answer. That'd obviously be cool.
Yeah. Well, thank you, everybody. I'm sorry we ran a little late.